to Bio 216, Chemistry of Carbohydrates, Lipids, and Nucleic Acids. In this video, we're going to look at the nucleic acid. We're going to concentrate on the nucleotide. Chemistry of nucleotides is our topic for this lesson. So, lesson you should be able to explain what a nucleotide is. List the components of nucleotide, describe the structure of different nucleotides, and list the role of nucleotides. Nucleotides are the monomeric units of nucleic acids. Nucleic acids are things you must have been hearing on the news and in different places. DNA is that we talk about. With DNA, you must have heard someone say he's going for a DNA test to see whether a child belongs to him or not. Or maybe they are carrying out a DNA test to find out whether a crime was committed by one person or the other. So that the DNA we're talking about is one of the nucleic acids we have. The next one is also the RNA. So what come together to form DNA and RNA is what we call nucleic acids. So remember in our last class we talked about nucleosides. So nucleotides are just nucleosides that have phosphoric acid esters attached to them. So when you have a nucleoside then linked to a phosphoric acid, it becomes a nucleotide. So what are we trying to say? That nucleotides contain nitrogenous bases, sugars, which are esterified to a phosphoric acid. That means which are joined to a phosphoric acid. Good. So, and this joining or this esterification can either be at position 2, 3, or 5 for ribose, and position 3 and 5 for deoxyribose. So, nucleotides are so important. Let me just give you. For every protein we have in our body, nucleotides come together in threes to form these proteins. And if there is any replacement, for example, if one of a nucleotide, okay, let me just use the alphabet. We have the G, we have the T, we have the A, we have the C. Good. And then which other one? Yeah. So they keep on, four of them keep on combining in different ways to form nucleotides in threes. So for something like hemoglobin, which is found in our blood, hemoglobin is the protein we find in our red blood cell. Hemoglobin is a protein that has two polypeptide chains. I'm sure you must have done this in your about 213. Good. In that hemoglobin contains it contains 147 amino acid units. And for the I'm talking about hemoglobin, the B chain of the hemoglobin. It has two chains. We call one A chain and B chain. The B chain contains 147 amino acid residues. And at position 6 of this amino acid residue, there is supposed to be a glutamic acid for the normal amino acid, the normal hemoglobin. Whereas for a sickle cell patient, that glutamic acid is replaced by valine, an amino acid called valine. And what happened? Glutamic acid is formed by a combination of nucleotides when GAG combine, GAG, they combine in threes. Whereas valine is formed by GTG. So the only thing that makes somebody to become a sickler is that that A in the middle is replaced with a D. Once this happens, all those things you see are caused just because of that. That's one nucleotide that is replaced by another nucleotide. That shows you how important nucleotides are. They combine, like I said, in threes to form amino acids. And of course, you know that amino acids are the ones that form. So, for example, if they don't combine well, form an enzyme, and different enzyme is formed, there will be a problem in the system. So that shows you how important nucleus carbon 5 of the pentose. You can see carbon 1, 2, 3, 4, 5. So it's to determine the name of this structure. So in this lesson, we have been able to say that nucleotides are phosphoesters of nucleotide. Nucleotides are phosphoesters of nucleosides, please. Nucleotides differ based on the nitrogenous base composition and pentose. Nucleotides could be mono, di, or tri phosphates. They have variety of roles in the body. So that's the end. Thank you for listening.